Okay, so hi everybody. So welcome to the second seminar of this week on the MaLaMoBi Seminar Series. So it's a great pleasure for me to introduce Professor James Wakeling. Uh, Several of you know his work, but just to introduce him, he is a professor at Simon Fraser University with an affiliate membership at the University of Victoria. So his main research interests are on muscles, and he had a PhD training at the University of Cambridge under Charlie Ellington, where he studied flight dynamics and performance of flight muscles. He had the postdoctoral work at University of St Andrews, and later on he worked also at University of Calgary. He spent some time at the Royal Veterinary College before joining Simon Fraser. So he has a lot of interesting work in many aspects of muscle physiology. And as a matter of fact, he has also an affiliation with the Department of Mathematics at Simon Fraser and the School of Engineering, I think. So he's gonna talk to us about his work on muscle design for optimal performance. So thanks a lot for the opportunity to hear from your research. And without further ado, I'll leave the stage to Professor Wakeling.
Well, thank you very much for inviting me to this seminar series. Oh, got to get this running. Okay. Why are some muscles pennate, but not others? Why do muscles have different types of muscle fiber? How does the additional mass and intramuscular fat that accumulates with obesity affect muscle quality? How does the additional muscle stiffness that develops during aging affect the way we move? These are questions I ask my undergraduate students. Some of the answers may seem obvious at first glance, some may not. These questions have been posed in the literature before, but the answers are not straightforward or even fully understood yet. In this talk, I'd like to introduce some concepts related to understanding muscle function and hopefully provide a toolkit of ideas by which we can reflect on these questions. So when muscle contracts, it will generate both a force and undergo a displacement, a change in length. And the mechanical work is the vector product of the force and the displacement. In order to understand the work done by the muscle, we need to know about both its displacement and the force that is generated. There are a range of methods which make it possible to measure muscle length and displacement. And some of these are sort of non-invasive, which are good for humans. So some methods include magnetic resonance imaging, where you can get the sort of 3D muscle shape. You can use B-mode ultrasound, where you can get the muscle belly and you can see the fascicles inside the muscle. In animals, you could implant sonar micrometry crystals, both to get sort of belly dimensions, but also fascicle lengths. And you could even essentially do laser diffraction through hypodermic needles in a device called an optical microendoscope. So we have a range of methods by which we could quantify something to do with the muscle length, but muscle force is not nearly so easy. A few studies have tried putting fiber optic cables through tendons. In animals, you can put strain gauges on the tendons, like E-shaped tendon buckles, but this is only done in a handful of labs around the world. Even if you're getting tendon forces, like the tendons are in series with muscles, but they're in series often with bundles of muscles. So you would need to dissect out or unbundle the tendons in order to get individual muscle force. So often you would get a common tendon force is not much better than inverse dynamics to estimate joint moments. And we don't really know much about directly measured muscle force. So I think it's fair to say that we don't know the muscle forces for most muscles in most species for most behaviors. And if a muscle force cannot be directly measured, then it needs to be estimated or modeled. So how do you model the muscle force? There's a couple of classes of muscle models. You could use a sort of mechanistic cross-bridge type model like a Huxley model that Andrew Huxley sort of introduced in 1957. This is on the top. You could have a Hill-type muscle model sort of stemming from his early works from 1938. The Hill-type muscle model is an empirical model just based on data that people have collected for force length and force velocity or properties of muscle. The hill-type model is computationally effective. And because of this, it's used for most biomechanics applications. For instance, if you were to use the sort of very commonly used OpenSim platform, that has hill-type muscle models embedded in it as the default muscle model. And this OpenSim has got over 250,000 downloads. So it's very common in biomechanics to be using hill-type muscle models, even if you don't realize that. And I think it would be fair to say that hill-type models are pretty much ubiquitous in biomechanics. But how good are the hill-type models? Should we use them? Should we trust them? I've got data here from a rat locomoting on a treadmill. We've measured the length of the fascicles with sonomicrometry. We got an EMG signal that I've put an envelope around the EMG signal. And then you can measure the force in black and predict the force from some kind of hill-type model, which we've got in red. What you find is that if you have very steady contractions and in situ experiments and looking at force velocity and force length kinds of contractions, hill-type models are pretty good. Like you get R-squareds of 0.8. If you're just looking at a sort of force length curve, you can get R-squareds of 0.99. But as soon as you look at in vivo contractions, where you have a naturally behaving animal or person, the muscle models get a lot worse. The R-squared will drop down to 0.4 or 0.5. This means that we can explain 50% of what's going on in the muscle force using the muscle model. But it also means that 50% of what's going on we cannot explain. So there's a large amount of our understanding which we currently don't have. Not only that, force predictions are worse for large muscles, rapid and unsteady movements, and submaximal activations. And I'd like you just to think about these points because we'll come back to that later. So what I'd like to do today is to explore different aspects of muscle contraction, of whole muscle contraction, that affect our understanding of muscle mechanics, and maybe piece together the parts of this 50% of muscle forces that we can't currently explain with standard hill-type muscle models. So I want to explore our ability to predict realistic forces. I'm going to break the talk down into 3 sections, which largely mirror some of the studies which have gone on in my lab. The first section will look at fiber types, recruitment, and energetic cost. Then we'll look at size, mass, and efficiency of muscle contraction. And then lastly, we'll look at 3D structure, energy flow, and mechanical work. So to start with, fiber types, recruitment, and energetic cost. I've got here a typical hill-type muscle model. It's got a contractile element, which would represent these sort of cross-bridge forces between the actins and myosins. It's got a parallel elastic element, which is where we have now sort of assigned the titin properties within sarcomeres. And there's a series elastic element, which is sort of largely composed of forces from things like the tendons within the muscle tendon unit. This could be a model for, let's say, a single fiber. You could also use it for a whole muscle, but if you want to go up to the size of a whole muscle, you obviously have larger forces than single fibers. So you just scale the forces up from this model. And that's literally all that you do. But the difficulty is our muscles have got many hundreds, thousands of muscle fibers, and the muscle fibers have different contractile properties. They can be largely classed into sort of into slow fibers and fast fibers. Slow fibers are type 1, fast fibers, there's a number of sort of subtypes. I've plotted here some of the mechanics which have been measured from human fast and slow fibers. We've got force velocity curve on the top, the type 1 slow fibers in red, type 2 fibers are in blue. You can see that the faster fibers will generate higher force, but more importantly, they will shorten out to higher shortening velocities. When you look at the power, the fast fibers generate more power, but more importantly, the velocity at which they generate greater power is a higher velocity, or their maximum power is higher velocity than for the slow fibers. We've also got the efficiency down at the bottom, and you see that fast fibers are generating their maximal efficiency at a higher velocity than the velocity at which the slow fibers generate their maximal efficiency. So the The mechanics of the different muscle fibers within the muscles are different, and it's going to affect our ability to predict realistic forces, will be affected by which muscle fibers are active. So which muscle fibers are active? We can start off with the commonly used sort of base plan for this, which is the size principle or orderly recruitment of different motor units. This was an idea that was first put forward in 1957 by Elrod Henneman, and it essentially says slowest motor units are recruited at low intensity contraction. Fastest motor units are recruited only at the highest intensity contractions, and therefore you would expect only slow motor units would be recruited for low intensity but fast contractions. Now, the difficulty is this may not make mechanical sense. Imagine that you're going down to a spin class in the gym. You might be pedaling really fast, your feet are spinning really fast, but if the intensity is low, if the resistance to the bike is low, you may not activate all of your muscle fibers, so you have to select which muscle fibers you would use. The size principle predicts that you'd use the slower fibers, but the mechanics would predict that the faster fibers would be better. So what's going on? Or why have we ended up in this situation that we've got our base plan as a size principle for motor recruitment? I think part of the reason is the way that motor recruitment was measured in some of the early studies. Now, what you would do is you would measure the EMG, which is the electrical signal to the muscle. You can either measure it with intramuscular EMG or with surface EMG. The EMG signal is the superposition of the individual motor unit action potentials from all of the motor units, which can be seen in the recording volume that the electrode is able to see. Now, there's a few difficulties here. If the muscle is really active, there'll be a lot of motor unit action potentials. The combined EMG signal will be very busy, and it'll be too busy, too hard to decompose all of the motor units out of a high intensity contraction. The other thing is that if the muscle starts to be dynamic, starts to shorten, then the muscle is going to change shape, it's going to bulge, the position and orientation of the motor units will change, and they'll change their position relative to the recording electrodes. This means that the recorded shape of the motor unit action potentials will change, and again, it makes it next to impossible to decompose the signal into individual motor unit action potentials. So because of this, the early studies on size principle looked at very slow isometric contractions at low levels of activity, and this is exactly the time when even the mechanics would predict that the slower motor units would be better. But we have done a range of studies trying to look at what happens for fast contractions. In order to do this, we need to have, So one thing that we can do to understand this is to get insight from some of the animal studies. And animal studies will commonly have looked at different muscles, not just looking at recruitment of motor units within a muscle, but they've recorded directly from different muscles. So an early study looking at this was the cat paw shake paper by Smith back in 1980. What they did was they had EMG recordings from the soleus muscle, which is almost 100% slow fibers. They also had recordings from the lateral gastrocnemius, which is a muscle which contains a much higher proportion of faster muscle fibers. During walking, which is slow, the cat used the slow soleus muscle, and there's hardly any activity in the faster gastrocnemius muscle. But during a paw shake, which is a rapid motion, the paw shake goes up to 10 hertz or 13 hertz, you see virtually no activity in the soleus muscle, but a large amount of activity in the faster gastrocnemius muscle. This would make mechanical sense. There's then a study from fish, Larry Rome, published in 1988. He titled it, Why Animals Have Different Fiber Types, and he was looking at the escape response of fish. Like if you scare a fish, it will do a sort of fast start escape response. It will swim off very quickly. And they showed that the speed the muscle fibers are shortening is actually too fast for the slow fibers to be any mechanical use, and suggesting that fast starts should only be powered by the fast fibers. Actual data from which fibers are used in fish came a bit later. Jane and Lauder published data from swimming in a bluegill sunfish. They have measured directly from either the red muscle which is in a sort of thin band down the side of the fish, or from the white fast muscle, which is the main bulk of the myotomal muscle. During slow swimming, it's just the slow muscle which is activated. At intermediate speed swimming, which is a graph in the middle, that you still have the slow muscle activated, and you additionally get some fast muscle is activated. And at fast swimming speeds, you get more fast muscle activated, but you start to see the slow muscles being turned off. Again, it looks like the animal has a preference for using the faster fibers for the faster speeds. So mechanically, it certainly seems that animals have got this right. What can we do for analyzing people? So here's a process that my lab has used. I've got EMG on the top. This is actually EMG from a cat, but it doesn't matter. It's the same principle. Each muscle fiber that you get will have, like it's bounded by the fiber membrane. The membrane has got a membrane potential. When you open up ion channels in the middle, you get depolarizations, and these will propagate along the fiber membrane and have a particular conduction velocity and a particular wavelength. And this results in the recorded signals that you get from your EMG system. So each action potential is a little wave. It can be characterized by its wavelength, conduction velocity, and thus frequency. And also note, each action potential occurs at a distinct time, is generating a characteristic frequency in the EMG. So if you can decompose the time frequency properties of the EMG sort of simultaneously, you might be able to get insight into the conduction velocity and the types of muscle fiber which are being active. We can do this with a wavelet decomposition. So if you have wavelets, I've got a wavelet or two wavelets here as a function of time. Wavelets are little waves. They have a time integral of 0, and they are well defined in time. If you look at a frequency representation of the wavelet, which I have on the graph on the bottom, you see that each wavelet is sort of a little frequency band. It's well defined in frequency. So we can use some of the properties of wavelets to help decode some of the information in the EMG signal. Once you do this, we then apply principal component techniques to classify the frequency spectrum. And wanting to skip over many years of work, we get to sort of graphs like this that can summarize the properties of the EMG signal. So this graph here is looking at the principal component loading scores. We've got PC1 on the vertical, PC2 on the horizontal. This is surface EMG in man, where we have different ways of eliciting different types of motor unit activity. What we find is that if you just get the faster motor units, you get a vector coming out to the right, which is the blue vector. If you have just the slow motor units, you get a vector coming out to the left. If you have all the motor units, like a mixed bag of motor units, you get a vector in the middle, which is this brown one, and it's actually higher up the graph because we have a higher intensity. We can do the same kind of thing by generating sort of simulated EMG using a volume conductor model, where we know exactly which motor units are active. We predict what the shape of the EMG would look like, and then we use this through our EMG processing system to see if we can get the same effects as we can experimentally see. Again, we do find the same effects. Fast motor units are this blue vector to the right, slow motor units are the red vector to the left, mixed motor units are slightly higher because we have more activity, and a vector, the brown vector, which is somewhere in between. We can also use this kind of technique onto some of our animal data. Here we've got 3 different muscles in the rat with a different fiber type proportion. The faster muscle, like with higher proportion of fast fibers, is medial gastrocnemius coming out to the right, slower muscle is on the left, and the mixed muscle is again in the middle. So we can take information which is in the EMG signal, decompose it to try to find which types of motor units are active, and then we can see what types of motor units are active for different types of muscle contraction. And when we do this, we can then build on the previous animal experiments which I've listed on the top. So the fish and cat examples I showed you showed that you would expect preferential activation of faster muscles between muscles, and this would occur for faster contractions, which involve high strain rates or high frequency contractions. When we now look at the recruitment of motor units within mixed muscle, we find again a preferential recruitment of the faster motor units associated with the high speed or high strain rate contractions. And we've got data now to show this for experiments on rats, goats, and in man. So what we can say is within our muscle, we have many thousands of motor units. They contain muscle fibers with different mechanical properties, and these will affect the mechanics, the forces that muscle can produce. And we need to understand which muscles are active in order to get the right kind of forces being produced. But we also need to use this information to drive our muscle models to predict those correct forces. So what we next went into was trying to use this information from the EMG to drive different types of muscle models. Here we've got an experiment. This is data from a goat. We have measured from the gastrocnemius. We've implanted EMG electrodes, implanted sonar micrometry crystals to get the fascicle length. We've got tendon buckles on the tendon to directly measure the muscle force. What we've done is we've used the data to drive standard heel-type muscle models which have the single contractile element, or slightly expanded models. I've called them two-compartment models, where we now have two contractile elements, one representing the sort of lumped effect of all the slow motor units, and one which is the lumped effect of all the faster motor units. What you can do, here we get the raw data, we get EMG on the top graphs. We can parse it down to the EMG contribution from the fast and slow fibers here in red and blue, calculate the activation patterns with some transfer functions. Again, we're getting activation states for both the fast and the slow fibers. We got the fascicle length from the sonar micrometry in the middle, measured force, and then we get the predicted force down at the bottom. The key things to note are that when we use the two-element model with the fast and slow fibers, and we drive it with the EMG that we've measured from the goats, we find in all cases we're getting higher R-squared, so better correlation, better predictions of the muscle force when we have our two-element model, which incorporates the properties of different fast and slow motor units. And then also on the right, we see that the errors between the predicted force and the measured force are reduced when you add in this extra element or you account for fast and slow fiber types within the muscle. So this is potentially good. This might explain a bit more of the unexplained force, but I was interested to know exactly why would the body do this, or how does the body do this? Like, it's easy to understand the size principle. If you have a low amount of drive to the spinal cord, you're just selecting the smallest motor neurons, which are going to be connected to the slowest motor units. But what might be the sort of signal driving the different recruitment that you get at different speeds? We thought at the time that it's related somehow to the metabolic cost of contraction. And because we showed that fast muscle fibers have a higher efficiency for fast speeds and slow muscle fibers have a higher efficiency for slow speeds. So what we've done is we've taken some human data. This is for cycling. This is an animation that comes out of OpenSim. The red lines in here indicate all the individual muscles which go into the model that we've got. Each of these red lines, each of these muscle models, we have tried and we have replaced with a two contractile element model with both fast and slow contractile properties. Now, what we're going to do is we predict what is the activation that you'd expect from fast and slow fibers if you optimize for a particular cost function. And the cost function we use is to minimize the metabolic cost of contractions. So within our muscle models, we've got similar force velocity, power velocity, efficiency graphs that I showed you before. We have specific properties for slow fibers in red, fast fibers in blue, and we end up with a sort of composite results down at the bottom. On the far right, you see results for essentially an isometric test. If you have an isometric ramped contraction, we'd see we've got a large red bar, which are a large contribution of slow motor units, and the faster motor units in blue doesn't have such a big contribution. When we look at cycling conditions, like which experimentally were at a low speed, like this is a low cadence here on the left, and they happen to have a high resistance or high torque as well, we find that again we get a higher amount of these slow fibers being recruited than the fast fibers being recruited. If you move across these set of conditions to the right, we're getting faster and faster contractions going at a higher and higher cadence, and the torque is also decreasing. So now on the right, we have the condition which has the high speed but low force contraction. And we find in this case, if you minimize the metabolic cost, the muscle is going to prefer to use a large proportion of fast motor units and a low proportion of slow motor units. Now, this is exactly what we have seen from the experiments for the fast-moving activities. So it would seem that if the body were to have a strategy of minimizing the metabolic cost, it will result in, during slow contractions, isometric contractions and slow cycling, there's sort of results which are consistent with the size principle, but the same rule, essentially the same cost function, will also end up in a result which prefers the faster fibers for the high-speed contractions, which is making mechanical sense and just what we see experimentally. So to try to tie all this up, a combination of experimental, modeling, and simulation studies have shown that motor unit recruitment patterns can vary. They're mechanically sensible, and they may result in minimizing the metabolic cost of contraction over a wide range of movement patterns. So we had these ideas coming from our experiments, and then there's a paper recently for a totally different purpose, but published in Nature Neuroscience in 2022, looking at macaque monkeys with recordings from both the cortical regions of the brain and EMG recordings. And in this study, they also found and showed that patterns of motor unit activity were surprisingly behavior dependent. It wasn't just locked into one orderly recruitment pattern. Now, this study didn't actually look at the mechanics of the contraction, but their results are coinciding nicely with the results that we're getting out from our lab. So all was looking good until Natalie Holt, when she was doing a postdoc with Andy Biono that I was working with, she got some results. I think she was trying to tease me with the title of this paper, saying the effect of fast and slow motor unit activation on whole muscle mechanical performance. The size principle may not pose a mechanical paradox. So this leads into the second part of the talk, and we're going to consider size, mass, and efficiency of contraction. So what was this study that Natalie Holt did? She was looking at rat plantaris muscle, and she was working out force velocity curves. We've got one here. This is actually a velocity force curve, but is the same data. When the muscle was fully active, like the 100% activation, the muscle shortens out to a maximum velocity of about 3.5 lengths per second. Then what you can do is you can give a submaximal activation to the muscle, but with the correct nervous stimulation protocol, you can either activate, largely slow muscle fibers or largely fast muscle fibers. I thought that if you just activated the faster muscle fibers, you would end up with a high speed. It would be at least as fast as if everything was active, possibly a bit faster. But the speed was not any faster than if you had just activated the slower muscle fibers. Like something wasn't quite fitting our framework. I was thinking that the muscle would contract at a faster speed, partly based on ideas which parallel what A.V. Hill published in a book he wrote titled First and Last Experiments in Muscle Mechanics, where he said that in a mixed muscle, Vmax, the maximum contraction speed of the muscle, would be that of the fastest fiber. But if you look at some of the literature more carefully, you find that Josephson and Edman in 1988 suggested that if you have inactive fibers within a bundle of muscle fibers, they may actually provide drag to slow the muscle down. And this is essentially the same that the Holt study found. So what Natalie Holt's study was essentially showing is that if you only activate 20% of the muscle, the whole muscle still has to shorten. So the 80% of the muscle fibers that are not active are having to be moved by the 20% that are active. And essentially, they might provide drag, they're dead weight, they're just not helping, even though they have to be shortened. So how can we consider this dead weight in the muscle? We need to somehow be able to model the effect of weight or mass within muscle. And there was a sort of clue to how we could do this. Michael Gunther published in 2012 a sort of enhanced Hill-type model that had muscle mass distributed throughout the tissue. In the Gunther study, they showed that the sort of inertial effect of this muscle mass resulted in delays in force development, but we were interested in how it can slow down the maximum speed of the muscle. So this is essentially Gunther's model. What we get here on the left, this is a sort of hill-type muscle actuator. It's got the contractile element and it's got a parallel elastic element. And there's a lot of these hill-type actuators in series along the muscle, but there are point masses distributed between them. So you can take the muscle mass, you can divide it up into all these different segments, distribute it down the muscle fiber, and on the sort of hill-type actuators, you can give it the hill-type muscle properties, force velocity and force length properties. You can then essentially wrap this up, think about this as a muscle fiber, and then you can use this as your muscle model to see now what the effect of mass is going to do to the muscle model. First of all, you could conceptually think about some of these things. The first thought you could have is what would happen if you scaled the model, geometric scaling. If you were to double every characteristic length, like double the length of the muscle, the cross-sectional area will go up as 2 squared, which is a factor of 4, and this will be proportional to the force. The volume will go up as 2 cubed, which is a factor of 8, which is proportional to the mass. So when you make the muscle bigger, the mass will increase faster than the force available to accelerate it. So maybe if you have any mass effect, the mass effect will be more pronounced for larger muscle. You could also think about decrease in activation. Here we get muscle of the same size, so the mass is the same, but if the activation goes down, the force will go down. And again, there'll be relatively more mass compared to the force available to accelerate it. So you may see a greater effect of the mass at low activation. These are two of the areas going back to the early study, the early slide, which the hill-type muscle models are not good at predicting force if you have large size and submaximal activation, by the way. So what we did was we took this sort of mass-enhanced muscle model, then we calculated essentially what the whole study would have said, and we got the results here on the right. We find that if you fully activate the muscle, you get a maximum shortening speed faster than if you only submaximally activate the muscle. There's something to do with the inertia of the mass and the fact that not all of the muscle fibers are active, or the muscle is not fully active, which is slowing down the muscle tissue. So then Stephanie Ross in my lab, she tried to experimentally actually parse out the mass effect. She had a rat plantaris muscle. She wanted to make the muscle heavier during the experiment, which is not easy to do, but she had a system that she could actually impose a virtual mass. So she had a little lever arm, there's a pin going into the muscle, and on the other end of the lever arm, she can add some weights. So the muscle will see the inertial effect of the weight on the far end of the arm. When she got the muscle to go through cyclic work loops, she found that the greater the mass that you impose on the muscle, the work decreased. I've also plotted one of her 3D model plots on right, which we'll talk about more later in the talk. But this also shows the same effect. If you increase the muscle size or increase the scale, and therefore increase the relative effect of the mass, you will see a decrease in the mass-specific work that the muscle can do. So it seems like the mass is producing some inertia within the muscle. There's going to be an energetic cost to moving this, but we wanted to know not just about the sort of energetics from the mechanical work, but what would this do to the metabolic cost of contraction and therefore the efficiency of contraction. So here we've got some tests with this one-dimensional mass-enhanced model. We're testing different size muscles, and we've got a size 10 and a size 100, which is bigger. We're putting them through cyclic work loops. And if you calculate the mechanical work, you see that the sort of mass specific or the proportional mechanical work goes down for the larger size, but the metabolic cost is much the same. And this means that the efficiency of the muscle will decrease. So we know that larger muscles will generate lower mechanical work at a lower efficiency than smaller muscles when they contract. Okay, so if we want to summarize this so far, we can say that mass effects are greatest at large sizes, low activations, and rapid movements, which is where the accelerations are highest. We want to know how big the effect is in people and whether it's relevant for looking at people. The test that we've done so far is essentially going into some musculoskeletal simulations on a bike. We've got muscle models where we've essentially increased the size of the model to increase the relative mass effect. And we find that at large sizes, you do get a significant mass effect. But at human sizes, it is significant, but the mass effect is only small. Now, I think the reason for this is that we're still using one-dimensional models in this test. And when the muscle shortens, for instance, it will increase in girth, it will increase in its thickness and its width. And so the mass is going to accelerate in all three dimensions. And just using a one-dimensional model is not capturing the full effect of the accelerations of the mass within the muscle. Nevertheless, we can now wrap up the two first parts of the talk. We find that muscle fiber and motor unit recruitment affect muscle force and power and the efficiency of contraction. And muscle mass also affects muscle force and power and the efficiency of contraction. However, just enhancing hill-type muscle models with fiber types and mass is not sufficient to explain all of this unexplained 50% of force that the hill-type models are unable to predict. So maybe we need to consider the problem from a different dimension. And this now leads us into the third part of the talk, which is going to look at the 3D structure, energy flow, and mechanical work within the muscle. So we started the talk by saying that muscles develop force and change in length, which means that they can do external work. The energy for this work is derived from the hydrolysis of ATP within the muscle during the cross-bridge cycle. But there are many stages, many parts within the muscle between the hydrolysis of ATP and the external work being done at the end of the muscle. And understanding how the energy flows between the cross bridges and the external work may give us insight into the design and use of muscles. So this is a figure that Chris Barkley has published in a few of his recent papers. It says you can take your ATP in the muscle, it will be hydrolyzed down to ATP and free phosphate, and you'll also get some enthalpy of contraction. This enthalpy will then be split up into both free energy and entropy. The free energy will also then be split into both cross-bridge work and heat of contraction. Now, supposedly, if you take the cross-bridge work and integrate it over all of the active cross-bridges, you should get the total mechanical energy available to the muscle during the contraction. This mechanical energy will be used for two things. It will be used to deform the muscle tissue and do sort of internal work within the muscle, which here we've called the internal strain energy potentials. And then some of the energy will result as external work that the muscle can do on the system does on the muscle. So if there is a finite amount of ATP being hydrolyzed, there'll be a finite amount of cross-bridge work, there'll be a finite amount of total mechanical energy. So any energy that's taken up as the internal strain energy potentials will not be available for the muscle to do external work. So understanding the deformations of the muscle and the internal strain energy potentials will maybe give us more insight into what's going on for the whole muscle contraction. So we can take our strain energy potentials. These are now actually represented as energy densities, which is the strain energy over the volume of the muscle, which is being considered. So we get our strain energy density within the muscle, and we can subdivide it into a few components. We get isochoric, volumetric, and kinetic energy densities. The kinetic energy density is something to do with the mass. Like kinetic energy is half mv squared. If your muscle has mass, then you'll get some kinetic energy density. The volumetric energy it could be considered the energy penalty to the muscle changing volume, is a sort of function which is going to try to keep the muscle at a constant volume. And then the isochoric component is everything within the muscle that's not involved with trying to maintain the same volume. You could sort of split this down into different tissues within the muscle tendon unit. You could look at the isochoric component within the muscle part or the tendinous part or the aponeurosis part. We'll focus now on the muscle part, subdivide this again into a cellular component. Imagine muscle fibers are actually cells, they're specialized cells with the contractile elements in. If you were to remove the contractile elements like the actins and myosins and titins, you'd just be left with a cell. And that cell has its own material properties. And this is the cellular component of the internal energy density. We also get ECM, the extracellular component. You could put in energy density for things like intramuscular fat, which we know also is going to affect the mechanics of contraction. And then we've got the fiber properties down at the bottom, which is what is going to represent the sort of cross-bridge level stuff between the actins and myosins. So the first three could be lumped together to call what we call the base material properties. And these have a particular stress stretch function and will determine how the tissue is going to deform. For the fiber properties, we can introduce stress stretch and stress strain rate functions, which are derived from the hill-type muscle model. We put in essentially our force strength and force velocity properties, which feed into this internal energy density for the fiber component. So what we see is that the contractile elements, which is down here as the energy density of the fiber, are only a small fraction of the total sort of energy content within the muscle. And so there'll be other things going on in the muscle which will affect the overall work that the muscle can do. So we can take this framework, we put this into a muscle model. Here we've got a sort of 3D schematic of a muscle. You can cut it up into many individual voxels. They are like each voxel is shown here on the top right. You've got the base material properties. It's a fiber reinforced composite biomaterial. So we've got the fiber properties, the contractile elements running through the base material. We can take a block of this muscle, which we've now illustrated in the bottom left. I've got this muscle undergoing a cyclic work loop, so it's being lengthened and shortened. You can see the muscle strain on the line graph on the right. We activate this muscle when it's shortening, and then we can calculate the internal strain energies, which is the bottom line graph. You can subdivide this into the strain energy potentials from the different components, active fiber, passive fiber, which is only a small component, and the base material. And you can see the energy flowing between the different components within the muscle as the muscle contracts. So we have this model, but is this doing the right stuff? Is it actually telling us how the whole muscle contracts? And for this, we need to go to a range of different experimental avenues to see if we trust the model. So how does muscle contract? Here I've got some data that we've measured from previous ultrasound studies. You see during a contraction on a pennate muscle, the fascicle length will decrease, the pennation angle will increase, the belly thickness changes, it depends on which muscle you're looking at. We find, or other studies have shown, that the intramuscular pressure increases during contraction. If you look at the shape of the fascicles in a muscle, you find that during contraction, they will curve into S-shaped paths. When we look at the outputs from the muscle model, we see exactly the same features. We get a decrease in the fascicle length, an increase in the pennation, an increase in the intramuscular pressure, and the fascicles will curve into S-shaped paths. Now, all of these features, they were not sort of encoded into the model. They are emerging from the physics of contraction and give us some confidence that the muscle model is doing the right things. We can also look at some results from other experimental tests. Here we get some realistic geometries. This is MRI tests from people doing low-level contraction, like passive or 20% contraction of their ankle plantar flexors. If you contract your gastrocnemius, you'll find some parts of the muscle are going to bulge out, some parts are going to get thinner. And we've got the bulging and the thinning of the muscle shown in red and blue. When you use a muscle model, you find similar regions of the model are bulging out and getting thinner. So the overall sort of 3D deformations on the surface of the muscle are showing a lot of common properties to what we actually find in an MRI test. You can look at the transverse work that the muscle does. Here we got data that Tobias Siebert's group has published, both from rat studies on the left, human studies on the right, but in common is if you provide a transverse load onto a muscle, like if you compress a muscle, then the force the muscle produces is going to decrease. Now, there's absolutely no way that you can predict this from a hill-type muscle model because those muscle models have no 3D properties. They have no transverse dimension. You cannot do anything in a transverse sense. But when we use our 3D muscle model, you find that if you put a transverse load onto the block of muscle, up here on the top, you see a decrease in the force that you get in the longitudinal direction, shown as this delta Fx. But we can now decompose it into what of the internal components within the muscle are contributing to this loss in force. And here we see there's a reduction in the volumetric component, which is contributing to this reduction in force. Here we're going back to the mouse experiments. This is the mouse experiment that Stephanie Ross did. When she puts these ideas and puts them into the 3D model, she finds that if you increase the mass in the model, you get a reduction in the work during cyclic contractions, but is now sort of parsed out into the different components of the internal strain energies. And here we got the kinetic strain energy density in this sort of cyan color. We have the active part of the fiber strain energy density in green. And the way that both these components change with size is shown here, and it's very similar in magnitude. So if we think that the active component of muscle is important, we must also think that the energy density and therefore the kinetic energy and therefore the mass is just as important within the muscle. So we have this 3D muscle model. It's showing lots of features which are very consistent with how real muscle actually works. So we can now use the muscle model to ask questions that we wouldn't have been able to address normally, either experimentally. And here is a question looking at the gearing within the muscle. So from a simple perspective, you could take your muscle fiber, which I've got here on the left. If you shorten the fiber, it has to increase in thickness to maintain its volume. If you have two fibers next to each other, then as they both increase in thickness, they'll overlap. They obviously can't both then be in the same space. And this is why the fibers will rotate to a steeper pronation in order to sort of maintain their spacing within the muscle belly. And when they rotate to a higher pronation, they'll actually shorten at a lower velocity. And this is the gearing effect within the muscle. So pennate muscle, the fibers will be geared, but we know that the gearing changes depending on the contractile conditions. There's been a fair number of animal studies looking into this, and they have showed that the gearing is dependent on the force that the muscle produces. And this is because the studies have maximally activated the muscle, and they have measured the gearing as a function of force. But we know that force and velocity are inversely related to each other. Now, I think functionally, it should be the velocity that's important. Remember back to the first part, if the muscle shortens too fast, then it won't be able to generate force. So particularly when the muscle is shortening very fast, this is when the fibers are in danger of not being able to produce force and when they most see the benefit of being slowed down. So I think the muscle should be geared the most when the velocity is highest. And we can address this now with our 3D muscle model. So we can take our block of muscle and then we can put it through a range of contractions and we can do this test. So first of all, we will stretch the muscle when the activation is low. We then activate it while it is stretched, and then we let the muscle shorten back to its initial length. At that point, we see what is the velocity of the fibers within the muscle and how does it compare to the velocity of the muscle belly, and therefore what is the gearing within the muscle. And we can do this increasing the muscle to different levels of activation. So we can look at a whole parameter space of different activations and different velocities. We find gearing results like this. I want you to focus on the graph on the top right. This is the sort of 3D surface plot. What you see is that if you hold the muscle velocity constant and sort of run along the muscle force axis, there is not much change in the gearing with muscle force. But if you hold the force constant and you now run along the velocity direction, you see that there is a large increase in gearing with increases in the muscle velocity, which kind of makes functional sense. And we have now been able to see this because we can test across a much bigger parameter space than it is possible to do experimentally. The last example I want to give you is a study on intramuscular fat. Because if the muscle has to do internal work to deform the tissue, anything in the tissue that needs deforming will take up internal work. And some of this might be the intramuscular fat. Now, every one of us has got some intramuscular fat. Even the leanest athlete has probably got 2% in intramuscular fat. But this, up to now, has always been ignored in muscle models. Because it's been ignored, there's no precedent for how to implement intramuscular fat. So we had to try a few alternative ways. So we tried a few models, shown in blue, where we introduce intramuscular fat. Sometimes we put fat in parallel with the fibers. Sometimes we'd replace bits of the fibers by the fat. Sometimes we had it distributed evenly within the muscle, but sometimes it was clumped into different parts. So we have different ways of introducing intramuscular fat, comparing it to no fat like lean models, which are shown here in brown. In all cases, if you have intramuscular fat, then you get a reduction in the muscle force and a reduction in the stress, which is the muscle quality. And the reduction in quality exceeds the amount of fat that you've put in. So if you put in 10% intramuscular fat, you get more than a 10% reduction in the muscle quality. And this is because of the extra work that's being taken up to deform the fat within the tissue. So trying to wrap up from all this, we can say for the 3D model findings, a significant proportion of cross-bridge work goes into deforming the muscle tissues. The internal cost of deforming the tissue detracts from the external work that a muscle can do, because there's only going to be a finite amount of energy available for the contraction. So because of this, muscle tissue properties have a profound effect on the mechanical output of the muscle. Then, where now? Trying to wrap up all three parts of this talk, we can sort of summarize this saying a significant proportion of unexplained force within the muscle models comes from the muscle tissue properties and not from the contractile elements. So understanding the tissue effects is going to be essential to understanding muscle function in patient-specific situations. We know that these tissue properties are important. If we continue to use muscle models without tissue properties, then we have little chance of understanding muscle function in health and disease. Incorporating tissue properties into muscle models is computationally complex. It's beyond the scope of many current musculoskeletal simulations that do require computational efficiency to work properly. But maybe we just need to be more pragmatic, and we need to triage the most important tissue properties which are relevant to the question that we want to address, and just try to incorporate those specific tissue properties into the simulations that we're trying to run. So we need to increase physiological and computational complexity of muscle models in musculoskeletal simulations to understand how muscle design influences movement. But just knowing how muscle functions is only the start of the process for understanding how muscle structure and function influence how we move in a principled and mechanistic way. We need to be integrating more complex muscle models into predictive simulations of movement in order to achieve this, in order to understand how changes to the muscle properties directly influence the movements that we do. And this is the area that my lab is currently focusing a lot of its attention. So I'd finally like to thank everyone for coming and logging into this talk. Special thanks to all the people listed here who've contributed to the ideas and the data that I've shown in the talk. And yeah, thank you for coming.
Thanks a lot to Professor Wakeling for the great presentation. So we have time for a few questions. I can ask, but I want to give the opportunity to the audience. Yes, Kathleen? Do you have a raised hand?
Hi, thanks. Thanks, James. That was a fantastic talk. I had a quick question about, you talked about the impact that neighboring muscle fibers have on the degree to which the muscles are able to bulge, or the fibers are able to bulge. I was wondering if you had any idea about the relative importance of neighboring muscles, whether they're active or not, in terms of how that's affecting the geometry of the contracting muscles or the forces other muscles are able to produce.
So if you compress on the muscle, the force goes down. And you might apply a compression, externally. It might be the bone is next to the muscle and it just can't bulge into the bone, so there's going to be external forces there. There's some experimental studies that the Herzog group has done in rabbit muscle, I think, where they individually activated all of the individually all the muscles within a muscle group, but then they activate them all together when now they're going to try to bulge and they're going to impinge on each other. And I think they found the sum of the forces isn't as big as if you added up all the individual forces because of these transverse actions, that if the muscle can't bulge, if it's being compressed, it's not going to be able to generate the force that it could otherwise do if it was isolated. So that's at the muscle level. You'd also expect at the fiber level, like the fibers are trying to bulge, they're being impinged by their neighbors from side to side. So it's all going to influence how the whole muscle contracts. Yeah.
Thank you.
Other questions? Well, I can ask one in the meantime. So maybe it's related to the earliest part of the talk where you introduced the two-compartment contractive model, ill-type model. And I
Yeah.
was wondering, so did you, so that must have a higher number of variables with respect to the one-compartment one. Is that correct?
Yes.
Yeah, so did you compare that model with a randomly chosen model, which include the model with one compartment, but has the same number of variables, the model with two compartments? Because of course, when you have, if you measure the accuracy based on root mean square error, which, if I remember correctly, is the one that you use there, well, of course, the models with a higher number of variables are going to have a facilitation. So when you increase the number of variables, the R square is going to increase no matter what.
Yeah.
Right. So that's a question. Yeah.
So no, we have not directly done this. Like one thing that you could do is you could make a more sophisticated muscle model with more parameters, and then you could essentially some parameters maybe you don't know, and you could just optimize the model to find the best parameters, and then you find the model has a better fit. That's opposite to the approach that we've done. Like when we've been doing these areas of work, we've tried to get experimentally determined values for all of the parameters as much as we can to put in. So when, yes, if you put in more parameters, then it's more likely that, or it's possible your model could have a better fit. But when we're not optimizing the parameters, we're still putting in all the data we're driving with is experimentally determined data.
Good. Awesome. Thank you. So other questions? So maybe you have another one like in the, so when you introduce, the earliest models, I think part one and part two probably, were kind of ODE models. When you go to ordinary differential equation models, when you introduce 3D models, when you introduce a spatial component, that the differential equation we described, that phenomenon starts to be complicated. So it means that it's going to be computationally expensive. So have you tried to include these 3D models of muscle to large scale musculoskeletal models?
I mean, this would be a great goal to have, and is obviously one which has occurred to me, that what we're trying at the moment is if you take musculoskeletal simulations of movement, like if you're using some of these predictive simulations of movement, we can sort of incrementally expand, make the muscle models more complicated. But to put in our whole 3D model within the framework of the predictive simulation is too much. So what we're currently trying to do is, like I was saying at the end, find out what the key parameters are in the 3D models and then try to emulate them in 1D, which obviously isn't as good as doing the complete 3D set. So we're running 1D and 3D models in parallel, learning what we can from the 3D models, and then trying to represent it in the computationally more efficient 1D models.
Awesome. Good. Very good. So, any last minute questions before we stop taking time out of Professor Wakeling's schedule? No? Good.
Thank you.